Hi there, boys and girls. I am Mr. Els. I am a teacher currently at Gr.12. The topic for this afternoon, genetics. During the discussion, I will focus on concepts and try to give an illustration to master them. Secondly, I will refer to complete dominance. Thirdly, incomplete dominance. Fourthly, codominance. And then in the last instance, sex-linked characteristics. Dealing with those, I would like to solve problems as we go along. I hope you enjoy the discussion. The first concept that I would like to deal with, the concept of a gene. You did protein synthesis, so it links up with this if I say to you that a gene is a small portion of a DNA coding for a particular characteristic. Now don't confuse yourself if I say to you, in this case we will deal, for example, eye color, or we will deal with the color of flowers. But there are genes on the same DNA molecule coding for the synthesis of a specific protein. So don't confuse those. It's relatively similar, if I can express it like that. If you have a look at the diagram on the screen, you will recognize the nucleus in which the nuclear material is found. The nuclear material, the so-called chromatin network, and on the onslaught of cell division, either mitotically or meiotically, the chromatin network will become visible as chromosomes, as illustrated on the screen over here. There you can see a chromosome, and the illustration tries to give us an indication that it consists out of a DNA molecule. There you can see the helical structure, but besides that, the diagram also gives an indication of a certain section, a certain portion, a certain fraction of the DNA molecule, which we then call a gene. Look at the next illustration as well. There you can see it. That highlighted section, that is a gene. And there are a number of genes on a specific chromosome. I will illustrate that later with the aid of a diagram. There, for example, you can see it is just a fraction of the nucleotides of a DNA molecule. There you can, for example, see on the left-hand side, adenine codes with thymine, thymine codes with adenine, or guanine with cytosine. That is basically the content of a gene. The second concept that I would like to refer to, alleles, or just the singular form allele. You find the phenomena that genes occupy identical positions on a pair of homologous chromosomes, controlling the same characteristic, but in different ways. Let me first deal with the locus before I go back to the concept of allele. The locus, that is the specific, the particular position of a gene on a specific or particular chromosome. The positions of genes remain constant. Here I have a diagrammatic representation just to show you. Here on the left-hand side, that is a representation of a chromosome. Remember, a chromosome consists out of two chromatids joined by a centromere. On the right-hand side of the screen, there you can see a homologous chromosome pair. I don't want to confuse you if I say the following in terms of the homologous chromosomes. A homologous chromosome pair, as illustrated on the screen, consists out of a chromosome of paternal origin, but it also contains a maternal chromosome. Bear that in mind. A pair of chromosomes or homologous chromosomes. Two chromosomes, one of paternal origin and one of maternal origin. If we go along and we continue with the representation, you will see that to the left and to the right respectively, each chromosome has been divided into certain segments. You can see here that that segment there is not the same size as that segment over there. And that one is not the same in size compared to that one. But if I go to the right-hand side, that specific segment and that specific segment is equal in size. That one corresponds with that one. You see, what I try to illustrate at this point in time is this. There's the chromosome. Each chromosome consists of a series, a number of genes. In other words, each of those genes, each of those segments represent a gene. And in the case of homologous chromosomes, you will find the phenomena that that segment on the one chromosome is equal in size compared to that segment on the opposing one. Bear that in mind in terms of if I return to the concept of alleles, genes occupying identical positions on a pair of homologous chromosomes, controlling the same characteristic but in different ways. It's a very important concept to master. I carry on with that because basically, to my mind, that forms the foundation of solving genetic problems. I went along and I colored each segment. That gene is red, that gene is red. That one is yellow, that one is yellow. So now you can see that the two yellow genes on the two chromosomes forming the pair, they occupy the same position. The same with the green ones, and eventually the same with the orange ones. But what is important, I would like to show you here, it may be that this specific gene, I'm pointing at that gene, the green one, you see it's darker in color, darker green, compared to the opposing one. That one is the allele of that one, and the vice versa. Boys and girls, it boils down to this, that this gene and this gene on two chromosomes occupying the same position, they determine the same characteristic. Let me give you an example. Let's say that gene determines the height of a plant. Then it simply means this, that that one there will also determine the height of the plant. They determine the same characteristic, but because I brought in two different colors, it shows that there are differences, and you have to bear that in mind. I'll come back to that later if I go to the genotype. What is meant by the concept genotype? It simply refers to the genetic makeup or the genetic composition of an organism. The phenotype. The phenotype is the physical appearance and the physiological characteristics of an organism, which of course is determined by the genotype. Don't forget that. The genetic composition determines what you see, because the phenotype, it's relatively easy to remember. It is what you see. You can see that the apple is red. you can see that the boy is tall, or you can see that the person walking in the street is very, very short. In other words, he is a dwarf. The phenotype, the physical appearance. But now we must get to what is meant by a dominant allele. The dominant allele refers to the fact that the allele or the gene whose characteristic always show up, appears in the phenotype, whether it's homozygous or whether it is heterozygous. And we will get to the two concepts, homozygous and heterozygous. Dominant allele, the characteristic, always show up in the homozygous state as well as the heterozygous state. But if we get to the recessive allele, you will see it shows up in a homozygous combination. In a heterozygous combination, it is masked, it's hidden. It's very, very important to bear in mind. The recessive characteristic, the recessive allele, the recessive gene becomes visible, phenotypically visible in a homozygous combination. But it becomes hidden or masked with the dominant allele in the heterozygous combination. You see, these things need explanation. So let's go to the concept of being heterozygous. Heterozygous, an individual which has two different alleles for a particular characteristic. Let's have a look. There is the pair of homologous chromosomes once again. You've seen that earlier. I take again the gene, let's say for this explanation, the so-called green gene. And you can see that there are different shades of green. It implies that, one, because they occupy the same position on the two chromosomes in the pair, they have to determine the same characteristic. And in this case, I used as an example height of plant. That is the characteristic. The gene there determines the height of the plant. The gene there determines the height of the plant. But now bear in mind that the height of a plant or the height of plants will differ. In this example, I say to you, let's take that this gene determines the height of the plant to be tall. The opposing allele or the opposing gene there determines the height to be short, a dwarfish growth form, in other words. You see, that is an individual. There are two different alleles for a particular characteristic. The particular characteristic, height of the plant. The two differences, or just the difference, the one determines the plant to grow tall. The opposing allele causes the plant to grow dwarfish, a short-growing plant. In the case of the concept being homozygous, it's again identical alleles for a particular characteristic. Let us explain that by using the diagram once again. I used exactly the same characteristic, namely height of plant. But now it is important to note that the gene indicated there determines the plant to be a tall growing plant. The opposing allele also determines the plant to be a tall growing plant. You see, if we go back to the definition, it says, or it states, two identical alleles for a particular characteristic. I repeat, the particular characteristic, the example that I use, height of plant or height of plants. The two identical alleles, in the first instance, both determine the plants to be tall growing. But you have to be very careful. Let's use the next slide. Can you see there? Again, the same characteristic, height of plant. But now, both determine short, dwarfish growing plants. This allele determines a short growing plant. The opposing allele determines a short growing plant. You see, if we go back to the definition using this specific diagram, it's exactly the same. It's homozygous. Two identical alleles for a particular characteristic. So make sure that you distinguish clearly between heterozygous, two different alleles for the same characteristic, as illustrated there. The one gene determines the plant to grow tall, the other gene determines the plant to grow short or dwarfish. In the case of being homozygous, they are identical. They determine the same characteristic, and the outcome in both cases will be tall. Or, as in this example also, the outcome may be short-growing plants or dwarfish plants. You see, up till now, I've made use of simple diagrams. I call them stick diagrams because you can draw them and you can make notes on them. There, where the cursor is at the moment, that is a gene. There is another gene. There is another gene. Consequently, a series of genes making up a chromosome. And the same concept is applicable to the chromosome being indicated at the moment. Each gene determines a specific characteristic. The outcome of that determination may be similar, as in this case, still on the screen, being short, or it may be different, as in this case, on the screen at the moment. Let us deal with complete dominance. I covered a number of important concepts. Complete dominance, what is meant by that? You will see that there are two descriptions for the same concept, the same principle. Let's have a look at the first slide. A genetic cross where the dominant allele masks the expression of a recessive allele in the heterozygous condition. Dominant allele masks the expression of a recessive allele in the heterozygous condition. You see, there's the concept of dominant, of recessive, of allele, of heterozygous, all in that specific statement. If we go to the next slide, dealing with complete dominance, I say to you that two individuals with pure breeding, contrasting characteristics cross to form offspring that all display the dominant characteristic. And then together with that, I say to you, in the heterozygous condition, the dominant allele expresses itself and masks the effect of the recessive allele. Let us have a look at complete dominance. If I say to you, true breeding, I hope you can follow my handwriting. True breeding. An organism is true breeding with regard to a specific characteristic. I don't prefer to say that the organism is true breeding because it's not true. It's true breeding with regard to a specific characteristic. This specific characteristic, that can again be, let's use the example, the height of the plant. I can refer to other specific characteristics. I can say the skin color of pumpkins. I can say the color of the petals of a flower. That is a specific characteristic, etc., etc. Now let's go to the point, what is then meant by true breeding? If I say it's true breeding for a specific characteristic, like the height of the plant, then I must qualify that. I must qualify that. Let us use the example, which is most probably in all the handbooks, Mendel's work, where he made a few experiments with garden peas. Remember what he observed. If I take a tall growing plant, it simply means if I say the tall growing plant is characteristic as far as height of plant is concerned, it means that they will always produce tall growing plants. I repeat, if I say a tall growing plant is true breeding for that specific characteristic, it implies that the offspring will always be tall growing plants. It will never ever deviate from that. Bear in mind, boys and girls, that I only work with one characteristic. In this case, I work with the height of the plant. So in terms of that specific characteristic, the plants were true breeding. But bear in mind, if I go along and I say to you, he also found, that is Mr. Mendel, also found that dwarf growing plants were also true breeding in terms of that specific characteristic. In other words, not all the plants that he experimented with were growing tall. Some plants only produced dwarfish plants, and they were true breeding as far as that specific characteristic is concerned. Again, it means that generation after generation, dwarf growing plants produced offspring that did not grow tall. They stayed dwarfish. Now, if we go along and we say then the following in terms of that, then I must say to you that this characteristic of being tall, the characteristic of being dwarf or short, bear in mind that's the phenotypic appearance of the tall growing plant and the dwarfish plant. That's the phenotype. I can see that they either grow tall or they grow short or dwarfish. But now it is important that, what about the genotype? What about the genetic composition of the tall growing plant and the genetic composition of the short growing plant? By now you know that the genotype of a specific characteristic is indicated by using alphabetical letters. Now we will leapfrog in this specific discussion if I say to you, in this example, I will use a capital T to represent the tall growing plant, and I will use a small t to represent the dwarfish plant. Because the tall growing plant was true breeding, then there are two capital T's, each actually representing the homologous chromosomes, and each representing a gene on the homologous chromosome. The same principle is applicable to the dwarf plant. So I repeat that if I say to you, to give an indication of the genotype of a plant, we will make use of alphabetical letters. And as I've said, in this case, I will use a capital letter to indicate the tall growing plant, and I will use a small letter to indicate the dwarf plant. You will see that in the case of the small letters, it's the corresponding small letters of the capital letters in use. Furthermore, I can go to the point in saying, seeing that we use capital letters, that simply indicates it represents the dominant allele. And seeing that we use the corresponding small letters, that will represent the recessive allele. Earlier, we made mention of the two concepts, the dominant allele and the recessive allele. But in using this format of illustrating the genotype, I would like to emphasize the following once again before I proceed. Let's use the two capital T's. What is the information that we can gather from that method of indicating the genotype of a specific characteristic? We can say, in the first instance, because I wrote it with capital letters, it represents the dominant allele. Secondly, because there are two letters, it represents the homologous. It represents the homologous chromosomes. And remember what I said initially, right at the beginning of the discussion. That one, for example, will be of paternal origin. That one will be, for example, from maternal origin. But then I also can say to you, because I give an indication of two, it represents the diploid. It represents the diploid number of chromosomes. So it is very important to note, what information can I gather from the genotype? Let us now proceed if I say to you, a true breeding tall growing plant was crossed with a true breeding short growing plant. I ended off by referring to the fact that the double capital T and the double small t represents the diploid number of chromosomes. Bear in mind that, and I'll try and indicate it like this, that meiosis will occur there, meiosis will occur there. So it means that from the diploid set of chromosomes, we will go to a haploid set of chromosomes. Because the process of meiosis reduces the number of chromosomes. Consequently, you find that during the process of meiosis, those two T's, because they are found on two different chromosomes, they will segregate. They will be found in different gametes. And the same with the genes, the chromosomes, as far as the dwarfish plant is concerned. And that's why I encircle them, to show that they are found in separate cells. That's just a personal taste. Now, what is important, as a result of meiosis, not only the diploid number of chromosomes are reduced, but you find that gametes are formed. These separate cells are gametes. So now you find the phenomena that another process must take place, or will take place, namely the process of fertilization. Fertilization, the fusion of gametes. You see, meiosis reduces the number of chromosomes. Fertilization restores the diploid number of chromosomes. And bear in mind that when gametes fuse, they all have the same opportunity, the same chance. That's why we refer to the chance of fertilization. You all know how to draw a Punnett diagram. A squarish block with 3 horizontal rows and 3 vertical columns. There's the one gamete, there's the other gamete, the gamete for dwarfishness, and that for being dwarfish as well. So those are the gametes indicated using this Punnett diagram. And bear in mind what I've just said, that this gamete has an opportunity to fuse with that gamete. That is a possibility. If that happens, you find the combination will be a capital T, small t. But it also has an opportunity to fuse with that one. And you will find that the same type of genetic combination is formed. But what is also important is the fact that this gamete over here can also fuse with this gamete over here. And you find this genetic composition. This gamete can also fuse with this gamete. You find the same genetic composition. So bear in mind, that gamete can fuse with either that one or that one. That is a possibility. There's a chance for that gamete or those gametes to fuse. And the same is applicable to the other one. And now you can see that from this, there I said initially, it's a true breeding plant for tallness. This is a true breeding plant for dwarfishness. And the offspring produced between a true breeding tall growing plant and a true breeding dwarf plant results into plants that will phenotypically all be tall growing plants. The offspring, phenotypically, they will all be tall growing plants. But bear in mind, look at the genetic composition once again. There's the genotype. There's the genotype. The genetic composition of this specific individual, it carries a gene for the characteristic of height of plants, determining it to become a tall growing plant. But it also carries a gene determining the height of the plant, but being a dwarf or a short growing plant. In this specific example, you will see again a capital T and a small t. The capital T, which represents a tall growing plant, therefore is the dominant gene, the dominant allele, whilst the small t is the recessive allele in this specific example. And that's why what we will see is determined by the dominant characteristic, the dominant gene or the dominant allele. That's why the offspring here will all be tall growing plants. But I repeat, take note of the fact that the genotype is a combination. That's why, therefore, we can say that all these individuals, they are heterozygous. They are heterozygous. Because now you can see that they determine the same characteristic, namely the height of the plant, but in different ways. The one gene determines the plant to grow tall. The opposing gene determines the plant to grow dwarfish. And that's why it's heterozygous. I said, in terms of complete dominance, it's a genetic cross where the dominant allele masks the expression of recessive allele in the heterozygous condition. I touched on dominant allele, I touched on recessive allele, and I touched on the heterozygous condition. There I said, two individuals with pure breeding contrasting characteristics. Pure breeding, it is equivalent to true breeding. Contrasting characteristics, the one determines the plant to be tall growing and the other determines the plant to be short growing. You see, it still boils down to the fact it deals with the height of the plant. And then, very important, the offspring displayed the dominant characteristic. That's why here I state again, in the heterozygous condition, the dominant allele expresses itself and masks the effect of the recessive allele. That's why I emphasized the genetic composition of the individuals formed. I would like to take it a step further if I the following. What will happen if we cross these heterozygous plants with one another? Phenotypically, both these plants are tall growing plants. But genotypically, in other words, their genetic composition contains a gene for tall growing plants as well as a gene for short growing plants. I would like to use the format that we would like to prescribe to you when you have to solve a problem like that. Bear in mind, they will pose a problem using words like homozygous, heterozygous, like being dominant. They will most probably give you an indication which letter to use. Must you use a letter R, or must you use a letter A, or whatever the case may be. Boys and girls, the principle or the principles stay exactly the same. In this case, I make use of the letter T, and I use the characteristic height of plants. You can substitute that with any other characteristic and any other alphabetical letter. Now in this case, I will say to you, let's say those two individuals are crossed. And I say to you that they're the parent generation, the P1 generation. The phenotype, this individual is tall. That individual is tall. The genotype, I gave it to you because I did not pose a specific problem in terms of a sentence or sentences and words. The genotype is that specific combination. The process of meiosis must take place. And I repeat that meiosis results into the formation of gametes, which means that during the process of meiosis, those two chromosomes will segregate, which means that they will be present. They will be found in different gametes. The same there. They will be found in different gametes. And as I've said earlier, that's why I encircle them. That's a personal habit. Good. Then the process of fertilization will follow. The process of fertilization will follow. And then I prefer and I stress, draw for yourself a relatively squarish block. There are 3 horizontal lines in that block, and there are 3 vertical columns in that block, the so-called Punnett diagram. Indicate one of the gametes there, there's another indication over there, there's another indication there, another indication over there. Sometimes we can say that those represent the male gametes, these represent the female gametes, or you can simply, as in my example that I use at the moment, you can just write in the word there, gametes, because there is no indication in this specific example of male and female. And then plain and simple, I say again, bear in mind that the chance of fertilization, this gamete has an opportunity to fuse with that gamete. And then you write the genetic composition in that block. But he has opportunity to fuse with this one as well. So there is that genetic composition. So that gamete can fuse with that gamete and you find this specific combination. Or that gamete can fuse with that gamete and you will find this combination. So in the first filial, the offspring from those two parents, I prefer to rewrite the genotype, and I write the word there, genotype, and the genotype is written like this. There is the genotype from the cross. And then the phenotype, I give an indication of the phenotype as well. That is a tall growing plant. That is a tall growing plant. That is a tall growing plant. And there is a short growing plant or a dwarf plant. But what is important as far as the illustration is concerned, or the problem is concerned, let's take this individual over here, there. I asked the same question I asked previously. What information can you gather from the genotype? Yes, it is diploid. It contains 2 full sets of chromosomes. Because I wrote it with 2 capital T's, it means that this individual is homozygous. Both the gametes, both the genes, they determine the same characteristic and they express it in exactly the same fashion. That one determines the height of the plant. That one determines the height of the plant. This one determines the height of the plant to be tall. This one determines the height of the plant to be tall. Therefore, it is a homozygous individual. A homozygous individual for a specific characteristic is therefore true breeding. I can say that that individual is true breeding with regard to the height of the plant. I qualify the specific characteristic. Those 2 individuals, they have the same genetic composition. Phenotypically, both are tall. How do I know that? That capital T tells me that is the dominant characteristic. That's the dominant allele. There, the dominant allele. Consequently, the small letter tells me it is the recessive allele. And there's the recessive allele again. So therefore, these 2 individuals, they are heterozygous. They're heterozygous. That's why I can't say to you that they are true breeding. These heterozygous individuals, they are hybrids. They are hybrids. That gene determines the plant to grow tall. That gene determines the plant to grow short or dwarfish. In the last instance, this individual over here, that one. You see, that individual is also homozygous. It's also homozygous. Therefore, for that specific characteristic, it's also true breeding. It's also true breeding. But look at the difference. Look at the difference between that individual and this individual. The capital letters indicate that it's homozygous, true breeding. The dominant alleles are present. Here, it's homozygous, but the recessive alleles are present. And you see, when recessive alleles are present in a homozygous state, it shows phenotypically. And that's why this individual is a dwarf plant. It grows short compared to the others. In conclusion, I specifically used a little bit of the information of Gregor Mendel's work. But here you can see from the phenotypic information that 3 of the plants are tall growing plants and only 1 is a dwarfish or a short growing plant. So that's why you find the phenomena that the phenotypic ratio is equal to 3:1. But if you go to the genotype, that one is homozygous tall, those 2 are heterozygous tall, and that one is homozygous short. So the genotypic ratio in this case will be 1:2:1. And that information of the phenotypic ratio and the genotypic ratio of being 1:2:1, it's very important to bear that in mind because it's clues, it's information that you require to solve certain problems. That is the discussion on complete dominance. I hope I made a few uncertainties clear. If there are any questions on this, please forward your questions and see whether we can give you a proper answer. Within a few minutes, we will go for a break. After the break, I will discuss incomplete dominance by referring to the definition, but I will make use of the diagrammatic representation illustrated on the screen. There's a similar one, and there's a third example. After the break, incomplete dominance. Dominance have been discussed. I will refer to codominance, and I will conclude the discussion by referring to sex-linked characteristics. Thank you.